Alright gang, here's our first video for chapter 20. This chapter is about DNA technology, and this is great. We're going to make a little bit of a shift here just for this last chapter of the semester, last unit of the semester, where we have been just learning how things in nature work, which is great, and that's like discovery science, like hey, there's something in nature that works and we're just going to discover how it works. And that's really fun. I do enjoy that science. But this chapter becomes not just how does it work, but what can we do with what we know, kind of thing. And this ends up getting pretty exciting, the things that we're doing. And this is technology is basically applying the knowledge that we've learned through science. And so this DNA technology is applying it to solve problems. Okay, so we're just going to go through a couple things in this chapter that scientists can very, very simply do with DNA to solve problems. Okay, and so one of the problems, the first one that we're going to look at is cloning a segment of DNA. Now, cloning, when people say that, students often think of like cloning an individual, like the first cloned sheep, Dolly, where they took an adult sheep, took some cells from it, and made it a genetically identical new sheep. And that is cloning. We're going to start a little bit more simple than that and just say cloning technically is just making an exact copy of some DNA. Okay, and so one of the things we can do is if we can take DNA and make a bunch of copies of it, especially if it's a specific gene, it allows us to do some cool things with the gene. It also allows us to do some good studying of that gene. Versus if we look at that individual gene in the midst of an entire genome of, say, a human being, it's really difficult to study because it's one little gene in billions of letters of DNA, and so it's really difficult. So, but if we can make multiple copies of a gene, now we can actually study it and we can do some things with it. So what I'm going to do is just jump right into kind of an overview of how cloning works. If they wanted to clone a gene, what would they do? And this overview should leave you with a ton of questions like, well, how would they do that? And how do they do this step? And so on. And we'll get to that in future videos. But so what we're going to start out with here is we actually have two different cells. This is a eukaryotic cell here, so we'll say it's a human cell, whatever. And then over here we've got a prokaryotic cell. And this bacterial cell here, it has a plasmid in it. Okay, and so one way that we can clone things, and there's lots of ways we can clone genes, but one way is to basically take a plasmid out of a bacterium, or a bunch of plasmids out of bacterium, but anyway, take plasmid out of a bacterium, and then take a gene, a segment of a gene out of the cell, and we're going to call it the gene of interest. So we'll talk about different genes of interest in a moment, but take a gene out of another cell, and basically insert it into the plasmid. And so now we have this plasmid, there's all the plasmid DNA, but we've inserted a new gene right here. And this is now recombinant DNA. Recombinant DNA just means that it is DNA that has come from more than one individual. Okay, so we have one type of DNA coming in here and a different type coming in here, and it's a recombination DNA, so recombinant DNA. Then if we can get that plasmid to go back into the bacterium, kind of like do a transformation type situation, go back into new bacterium, and so we will have bacterium that has this cloned, has this recombinant DNA in it. And so as this bacteria makes more and more bacteria, every time they go through replication and then binary fission, they're going to make a new plasmid that has that gene of interest in it. We can do a couple things with this. Okay, so one thing we can do with this is as these bacteria keep replicating, we're getting more and more copies of the gene, and you can actually extract that gene. You can kill these bacteria, lyse these bacteria, and extract that gene from the group, and actually end up do some interesting things. So one, you could just research it, but things they've done is they've taken down, here's a pest control gene, and what they did is they found that maybe soybean, or anyway, maybe one type of plant is resistant to a certain type of insects, and literally it's a chemical that they produce that keeps the insects away. So if they could figure out which gene it is that makes that chemical and make multiple copies of that gene, maybe they could put it into, in this case, a corn plant so that insects wouldn't attack that corn. Okay, and they've done this. This is something they've done. Actually, if you've heard of the whole GMO, genetically modified organism thing, a lot of the corn and wheat and soybeans, a lot of that stuff is genetically modified to prevent pests from attacking or to enable them to resist other types of situations. Anyway, so there's that. You can read the book for other examples. But that's one thing we can do is just make copies of the gene and try to insert them into something else. Okay. The other thing we can do with these clones of the gene, these bacteria that have clones of the gene in them, is we can just let these bacteria do what they do. And what bacteria do is they read the DNA that's inside of them and they make the proteins that are present. So if we can get a useful protein, and there's a couple examples here, for instance, human growth hormone, HGH. Human growth hormone, if you had an individual who could not produce their own human growth hormone, they wouldn't grow very much. They'd have stunted growth. Well, if you could get bacteria to produce that protein, human growth protein, the human growth hormone, you could basically have these bacteria make a lot of that and take it and inject it into a person who's low on human growth hormone, and all of a sudden they'd be able to grow. Okay, and that would be pretty cool. And so these are things that they are doing. Okay, now what we need to do is learn the process of how do they do this whole thing. How do they get genes out of the eukaryote here? How do they get the plasmid out? All these kind of things. We need to learn all those steps. So the first thing we have to learn about is kind of a step before you can actually take a gene out of the gene of interest, before you can find the gene of interest. And that'll be the next slide here. So the first process to understand, if we're going to figure out this whole cloning inside of a bacteria thing, is what a restriction enzyme is. Okay, and first of all, restriction enzymes are enzymes that are found in bacteria, and what they do is they just chop up DNA, and they are a defense mechanism for bacteria. These enzymes are in there, and if a virus were to infect a bacteria, the bacteria can chop up the viral DNA, and then it won't do its job, and the bacteria is fine then. The way the bacteria keeps from having their DNA chopped is they methylate, so they put methyl groups on their DNA, on the bacterial DNA, and then it is safe from these restriction enzymes. A lot of viruses don't have that methylating happening, and so the restriction enzymes don't work on those. But anyway, so restriction enzymes chop up DNA. The way they work, restriction enzymes, is if you were to put a restriction enzyme in a solution with just a bunch of DNA, those enzymes will basically scan the DNA, so they can scan along the DNA until they get to a specific sequence that we call a restriction site. Okay, and it would be specific for every different type of restriction enzyme. There's dozens of types of restriction enzymes. And so whichever one we're talking about right now has a specific sequence that it's looking for, and if I read in the 5' to 3' direction on the top strand, it's GAATTC. Okay? If I look at the bottom strand and read 5' to 3', it's the same sequence, GAATTC. So it reads the same on both strands, and basically this enzyme looks for this sequence. When it finds it, it chops the DNA. Okay? The way it chops it is uneven. It does not chop the DNA straight down through the middle of the restriction site. Instead, it's a little bit of an uneven chopping. Okay, and in that uneven chopping, it creates what we call a sticky end. Sticky end, where one strand on the DNA is short, the other strand is longer, and I can see the opposite happening here, a short strand and a long strand. The reason they're called sticky is if I just cut this DNA apart and then let it sit for a while, chances are that these A's and T's, they're going to find each other and they'll just stick back together. That's hydrogen bonding. They stick back together. Okay, so these sticky ends, they can be useful if we wanted to insert something in between here and here. What we could do is cut our green DNA, okay, with our restriction enzyme, and then take what we call restriction fragments. If we take a long piece of DNA, so we got a long piece of DNA here, and we put a restriction enzyme in with that, it may find this specific sequence, I don't know, 100 times along this strand of DNA, but we'll just say a couple. And so it's going to cut the DNA every time it finds that specific sequence, which would be a bunch. Okay, so it's making all these little fragments, fragment, all these little fragments. Those are called restriction fragments. Okay, well, if we wanted to take one of these, we'll say this one, and put it inside of this sequence, okay, what we can do is if we use the same restriction enzyme to cut here and here, okay, I'm going to have sticky ends, just like I have on the green chromosome. So I have my gray chromosome down here with sticky ends that match the green one. And so there's a chance that if I have a solution full of the green chromosomes and full of the gray restriction fragment, that they will end up, the restriction fragment will end up inserting itself into the green chromosome. Okay, and that's what we have happening right here. Okay, last thing that would have to happen here is we have to put DNA ligase in there. If you remember, ligase is what, if you remember the Okazaki fragments from DNA replication, DNA ligase is what connected those fragments back together. So we see this happening here. Okay, here they're in fragments. There's little spots between the G and the A right here, and now those spots aren't there, that empty space. So the ligase puts that back together. But that's what a restriction enzyme does. Now we're gonna, I'm actually gonna end this video now, and in the next video we're gonna look at how we can use this to clone DNA within a bacteria.